Good morning, everyone. I'm Dr. Santosh. Today we will be talking about fatty acid oxidation. In fatty acid oxidation, we will first talk about main pathway, mitochondrial pathway. So, fatty acid oxidation is divided into the major fatty acid oxidation occurs through beta oxidation, and that beta oxidation of fatty acid. This beta oxidation is mitochondrial beta oxidation. Mitochondrial beta oxidation. So this is major pathway, and then minor pathway. There are minor pathway or alternative pathways. There are minor pathways. So we will talk about this minor pathway or alternative pathway later. Alternative pathway. So first we will mainly focus on beta oxidation of fatty acid. We will talk a little bit about this only. So beta oxidation, mitochondrial beta oxidation. We will put some questions for this pathway. First is, when does it happen? When does this pathway happen? So this pathway happens during fasting condition. Fasting condition. So always in fasting condition, because the fuel in fed condition, main fuel in fed condition is glucose, and in fasting condition the main fuel becomes fatty acid. So fasting and starvation or prolonged fasting condition, starvation. So this is the answer. Now we'll put why question. Why does this pathway happens? Obviously to provide energy. We all know the fat is storage form of energy. So energy, it provides energy to body and for gluconeogenesis gluconeogenesis. Now third question we ask is, where does this happen? It happens inside mitochondria of nucleated cells, and that too oxidation of fatty acid itself happens in almost all nucleated cells except brain. So we'll talk why it does not happen in brain. So now next question is, what are the things that take part in reaction? So rate limiting step or rate limiting enzyme is CPT1 at the tissue. CPT1. What is this CPT1? Means carnitine palmitoyl transferase 1. It is present in outer mitochondrial membrane. Now, so in adipocytes, this pathway is also regulated at adipocyte during lipolysis. So we will talk about this also. These are the two steps where it is regulated, at lipolysis and at the entry of fatty acid into mitochondria. It is done at CPT1. Now, what are the vitamins? So we will always focus on, whenever we talk about the pathway, we will always talk about vitamins that are involved in the pathway. So vitamins involved in this pathway are B2, B3 and B5. So vitamin B2 is in the form of FAD. Vitamin B3 is in the form of NADP. And vitamin B5 is in form of coenzyme A. So these things will be required. Not NADP. It is NADP. NADP is required in fatty acid synthesis. So NAD, FAD, and then coenzyme A. Now, how does it happen? So we will talk about this in a bit detail next. So where does it happen? So we we are answering here the question, where does it happen? Fasting and starvation, that we have already done. And then so fasting, let me talk about this fasting and starvation. So when we talk about fasting and starvation, so biochemically fasting and starvation can be defined by this molecules glucagon. There are some other hormones also, epinephrine. So we'll talk only about glucagon. So everything will be done with the glucagon only. If you understand glucagon, you will understand every other thing also. Glucagon, what does it means? When glucagon in concentration in blood rises, so what will happen? CAMP concentration will increase. And then if CAMP concentration increases, and that means intracellular phosphorylation. How? So protein kinase, because protein kinase A. So protein kinase A will increase because protein kinase A is also called CAMP dependent kinase. CAMP will activate protein kinase A. Now what will happen next? So this protein kinase A will cause phosphorylation of all protein. Phosphorylation of proteins. So this is very important fact, will require this. Now to up during fasting condition, so it is just remember fasting condition is defined by glucagon, number 2 point glucagon, then third is CAMP, fourth is protein kinase A, and then protein kinase A means phosphorylation. So this is the thing that defines, these are the molecules, these are the state that defines fasting condition, fasting and starvation. Now let me talk also about fed condition. So fed condition is defined by insulin, and everything opposite, low CAMP, MP, here protein phosphatase, and then dephosphorylation. So these are the defining. Now we are talking about fasting condition only. So fasting condition, what will happen? Dephosphorylation will require this. So that is why I have written here, will require this. Now fasting condition, what will happen? So during fasting condition, glucagon is high. This glucagon activates fatty acid oxidation. Now, fatty acid oxidation happens in almost all tissues. So, major tissue in our body is liver and muscle, skeletal muscle and cardiac muscle. So, this tissue utilizes fatty acid in very high quantity. And renal cortex, not medulla, renal cortex. Renal medulla always utilizes glucose. So, renal cortex, cardiac muscle, both muscle, cardiac and skeletal, liver, they utilize fatty acid during fasting condition. Now the fasting during fasting condition, brain does not utilize, or in starvation also why brain cannot utilize, one of the reason brain cannot utilize fatty acid is this fatty acid are big molecule that cannot cross blood brain barrier, so that is why this fatty acid oxidation cannot occur in brain. One reason. Second reason, maybe there is not such enzymes, maybe that could be a reason. One of the reason is the brain does not have this fatty acid, cannot pass blood brain barrier. So mitochondria of all nucleated cell oxidizes fatty acid, and it does not occur in brain. That's very big point. Now what does brain utilizes? Ketone bodies. That it is like fast food, it is like processed food. Ketone bodies are processed fat, processed fat, and this ketone body are synthesized by processed fat by liver. So liver synthesizes this ketone body and provides to brain during starvation condition. Now let me talk about what are the facts. So ATP is required, carnitine is required, fatty acid obviously, and then these three vitamins, as I told you, vitamin B3, vitamin B2 and vitamin B5. Now Let me little bit talk about this alternative pathway before going to measure this mitochondrial beta oxidation. So this alternative pathway is of three types. These are very minor pathways, and not all molecules. Maybe 95% of the fat is oxidized through our beta oxidation. That is mitochondrial beta oxidation. So this alternative pathway is of two types: alpha alpha oxidation, and then this is peroxisomal. You can write it peroxisomal. This is also peroxisomal. So it does not happen in mitochondria. Peroxisomal alpha oxidation, peroxisomal beta oxidation, and ER, endoplasmic reticulum, omega oxidation. So peroxisomal beta oxidation, peroxisomal alpha oxidation, and endoplasmic reticulum omega oxidation. So this three reaction occurs outside the mitochondria, two in the peroxisome and one in the endoplasmic reticulum. So only one fatty acid is oxidized through alpha oxidation, that is called phytanic acid. This is very important. We will talk about this later. And then a very long chain fatty acid is oxidized through beta oxidation. Through this peroxisomal beta oxidation, peroxisomal beta oxidation, this long chain fatty acid, very long chain fatty acid converts into long chain fatty acid. And this long chain fatty acid will join normal, that is mitochondrial beta oxidation. So these are the alternative pathways or minor pathway in nutshell. So we will talk about this minor pathway later in detail. So in this class we will talk about beta oxidation only. So beta oxidation, let me talk about beta oxidation. Beta oxidation occurs during, as I told you, it happens during fasting condition. So we will talk in context of hepatocytes, liver. So here we are taking the context of hepatocyte, beta oxidation of hepatocyte. So what will happen? So hepatocyte does not have store of fatty acid, that during fasting in adipocyte it breaks into fatty acid and glycerol, and this is done by glucagon. This breakdown is done by glucagon. So glucagon, because of glucagon, I told you, increase CAMP. Then this CAMP increases hormone sensitive lipase. Remember, this is very important enzyme. This enzyme is being phosphorylated by this. So it gets phosphorylated and activated. So this active enzyme will do lipolysis in adipocyte. This events happen in adipocytes. So in adipose tissue, or triglyceride will convert into fatty acid and glycerol. So what is the major fatty acid in our cell? Is long chain fatty acid is the main fatty acid. Long chain fatty acid. What are those long chain fatty acid? One long chain fatty acid is palmitic acid. There's 16 carbon palmitic acid, and then second is 18 carbon stearic acid. This palmitic acid and stearic acid, long chain fatty acid, will have to enter into mitochondria. Now this, after this lipolysis, these fatty acids and glycerol, they will enter blood, and through blood they will enter the mitochondria of the hepatocytes. Now let's see. So when they enter the mitochondria of hepatocytes, these all fatty acids will be converted into fatty acyl coenzyme. This step is called activation of fatty acid. Now this is the only step where ATP is utilized. This is the only step where ATP is utilized. So ATP converts here into AMP. That means 2 ATP. This equivalent to 2 ATP. Utilization of 2 ATP. That you can see the similar reaction you can see in amino acid activation during translation. So what happens? So activation of fatty acid. So fatty acid converts into acyl coenzyme A. Now this acyl coenzyme A will enter into mitochondria. Now But there is one problem with this acyl coenzyme A. This long chain fatty acyl coenzyme A. Let's be precise. Long chain acyl coenzyme A. It will enter mitochondria, but it cannot enter mitochondria like that. So it will have to convert into it will. So obviously this acyl coenzyme will go under beta oxidation and it will produce acetyl coenzyme A and then FADH2 and NADH. So we will talk about this later. First, let's talk about acyl coenzyme. How acyl coenzyme, a long chain fatty acyl coenzyme A enters this mitochondria. It cannot enter mitochondria like that. So it will have to be converted into one molecule called acyl carnitine. So this acyl coenzyme A is replaced by carnitine. So carnitine is very important here. So when long chain fatty acid binds with carnitine, then long chain fatty acid, then only it can enter mitochondria, and this now acyl it will enter mitochondria, and in mitochondria it will again change into previous form acyl coenzyme, and now acyl coenzyme will undergo beta oxidation. It will produce acetyl coenzyme A plus FADH2 and NADH2. This is the whole process. This is the most important slide of the beta oxidation. If you remember this also, you can remember whole context of fatty acid oxidation, especially mitochondrial fatty acid oxidation. So most important step is lipolysis. One of the very important step is lipolysis. Then second most important is step is transport. So transport is regulated by CPT1, and this is also regulated by glucagon. So these are the most important step of fatty acid oxidation. Let's say fatty acid oxidation. When we talk about the fatty acid oxidation, it also includes lipolysis as well as beta oxidation. Now fatty acid oxidation, so it happens through four stages: lipolysis that we have shown here, and the activation of this fatty acid, that is here activation. Then mitochondrial transport, third step is mitochondrial transport. Different proteins are there involved in mitochondrial transport. Most important is CPT1, CPT2 and CAT protein. So CPT1 is carnitine palmitoyl transferase. So this is the protein that converts this CPT acyl coenzyme A into acyl carnitine, and this is the protein, this converts this acyl carnitine back to, this is the protein CPT. This is the function of CPT2. This is the function of CPT1. They are present in mitochondrial membrane. This is present in outer mitochondrial membrane, and this is present in inner mitochondrial membrane. Inner. And this CAT, this translocate, this is the protein. So this protein, this CAT will transfer this acyl carnitine from outside to inside. So these are the function of these three proteins. So CPT1 is the most important protein here, that transfers. CPT2 is also important. We will talk about the disease of the CPT2. And CAT, carnitine acyl translocase. This is the translocase. These are transferase, and this is translocase because it transfers acyl carnitine from outside to inside. And that is all about this beta oxidation. Now let's talk about all about the fatty acid oxidation, and then let's talk about this beta oxidation precisely. So what happens in beta oxidation? So this is the very important point. The only step that requires fatty ATP is activation of fatty acid. So this is the only step where 2 ATP is utilized. The same thing you will see in amino acid activation. So now activates long chain fatty acid. So let me differentiate between long chain and short chain fatty acid. So long chain fatty acid, always long chain fatty acid gets require carnitine. Only long chain fatty acid require, only long chain fatty acid require carnitine. A very long chain fatty acid has to convert into long chain. That happens in mitochondria. Otherwise, it cannot enter. So, very long chain fat, only long chain fatty acid require carnitine. So, long chain fatty acid, it is between, very simple. It's 12 to, just remember this number. Put this 2 in front and 1. 12 to 21. This is the rough like number 1 2 to 2 carbon. So this the carbon, the fatty acid which have carbon in between are called long chain fatty acid. Below this is called short chain fatty acid. Short chain. Above this is called very long chain fatty acid. So just remember this important fact. So only long chain fatty acid require carnitine. Now this is beta oxidation happens. Beta oxidation means the oxidation of carbon at beta carbon. So this is alpha carbon, the carbon attached to functional group. This is called alpha carbon, and this is beta carbon. Oxidation of this carbon occurs, and because of that, bond will be broken like this. So bond will be broken. So bond will be broken between alpha and beta carbon. So this is what happens in beta oxidation. Beta oxidation is nothing but breaking of bond between alpha carbon and beta carbon of fatty acid chain. So this is the fatty acid. This is the palmitic acid. Now palmitic acid has, you can see this is carboxylic acid, and then palmitoyl coenzyme A, and then these are the bonds between this palmitic acid that were formed during palmitic acid during fatty acid synthesis. So during fatty acid synthesis, bond are formed like this. So this bond will be broken down. So this is alpha carbon, this is beta carbon. Again, when it breaks, this become alpha carbon, this becomes beta carbon. Again, when these bonds break, this become alpha, this becomes like that. So beta oxidation, let's see. So this bond breaks, that means this is alpha carbon. Bond is broken between alpha and beta, and because of that, what will happen? So new acyl 14 carbon acyl coenzyme will be formed. Again, bond will be broken between alpha and beta, and then 12 carbon acyl coenzyme A is formed. Now this bond is again broken, and this forms 10 carbon acyl coenzyme A. When again this bond will be broken, and then this will form 8 carbon, and it goes like this only. So this way you can see acetyl coenzyme is being produced because of this bond breaking. How many acetyl coenzyme from one palmitic acid? Total is 8. Total 8 acetyl coenzyme A from acyl. That's palmitic coenzyme. Palmitoyl means we can write is acyl also. So in common word, we call it acyl. All long chain fatty acid, we call it acyl. So acyl means this palmitoyl or stearyl coenzyme A, we call them like that. So palmitoyl coenzyme A will convert into 8 acetyl coenzyme A, and this 8 acetyl coenzyme A from will have 7 bonds. 1 2 3 4 5 6 7 bonds, and these 7 bonds will be broken down through oxidation to oxidation, OO. So OHOT reaction. So 4-stage reaction will break the bond. Again 4-stage reaction will break this bond, so forth. It happens so forth. 4-step reaction will break these bonds. This break these bonds. These bonds uh, require two oxidation reaction. So OO, OHOT. So O for oxidation, and this O also is oxidation. Oxidation, hydration, so addition of water, then oxidation, and thiolase reaction. Thiolase means bond breaking reaction. So O hot reaction. This is, remember this, O hot reaction. This O hot reaction happens at all bonds. Because of this reaction, so there will be production of one FADH2. The first oxidation will produce one FADH2, and then second oxidation will produce one NADH. Whenever oxidation happens, there will be release of energy. It is general rule. So here is a release of one FADH2. It is equivalent, energy equivalent, and one NADH. So from each bond, all bonds, so 7 bonds are there. From all these 7 bonds, there are generation of 7 FADH2 and 7 NADH2. So one FADH means how many? One FADH means is equal to 1.5 ATP, and then one NADH is equal to 2.5 ATP. So total will be 4 ATP production from each bond. So 4 ATP production will be from the each bond, and total ATP will be 28 ATP production will be from this 7 bond of beta oxidation. So beta oxidation occurs at the beta carbon, and because of that bond lysis happens. So we can take it as beta oxidation is generally breaking of bond and formation of acetyl coenzyme, and each bond, there are 7 bonds in palmitic, each bond will produce 4. So total is 28 ATP production here. So this like that. So 7 7 21 O for oxidation, and you can see the name of enzyme, fatty acyl coenzyme dehydrogenase, and this also is a dehydrogenase. Beta hydroxyacyl coenzyme dehydrogenase. Enol beta hydrolase is this enzyme, and then thiolase is the last enzyme. So just remember this mechanism. What happens? This beta oxidation is breaking of bonds. Just remember that 7 bonds are there. 7 bond. Each bond break is release one FADH2, one FADH2, one NADH, and then equivalent to this FADH and NADH is production of 4 ATP. 4 7 ja 28 ATP will be produced because of this. Now, if you can remember, 2 ATP is utilized in the beginning. So here net ATP production is 26 ATP. So ATP is utilized for the activation of fatty acid. The only step that requires ATP. Now let's talk about this. So see this step. This is O hot step. So first step you can see is release of FADH2. So this is done by this enzyme, acyl coenzyme dehydrogenase enzyme. Just remember dehydrogenase. You don't have to remember all enzymes. These are not that, but it's very easy. Acyl coenzyme that was undergoing oxidation. This dehydrogenase of acyl coenzyme dehydrogenase. Now it will produce FADH. This is oxidation, and then hydration, addition of water. Hydratase is addition of water. So this is in oil coenzyme hydratase. So addition of water is there, OH and H, double bond is gone, and then so oxidation again will produce keto group. So this is beta hydroxyacyl coenzyme, again dehydrogenase. Remember, third is also dehydrogenase. It is oxidation step, and the fourth is thiolase, breaking of bond. So this step, this 4 step are involved in cycle. This 4 step goes in cycle. So this is how beta oxidation happens. Now let me summarize beta oxidation. So palmitic or palmitoyl coenzyme A will require 7 coenzyme A. This is vitamin B5. Vitamin B5, don't forget that. So many vitamin B5 is required in high quantity during beta oxidation. Then 7 FADH is required, 7 NADH is required, and then production is 7 FADH, 7 NADH, and then 7 proton is released out of 7 FADH2 and 7 NADH and 7 H plus. So this 7 production breaking of 7 bonds. So this 7 bond will release. So 28 minus 2 ATP is 26 ATP. So there's 8 acetyl coenzyme A. Each acetyl coenzyme A will produce, if this all fatty acid has to go under beta oxidation, then it will produce 80 ATP. 80 ATP, and this ATP plus 80 plus 26 equals 106 ATP will be produced. The total ATP produced from palmitoyl coenzyme is 100 plus 6. But what happens in liver? Liver only beta oxidation happens. So no further oxidation of acetyl, only beta oxidation of fatty acid happens. No further oxidation of acetyl. Acetyl coenzyme A will not go under oxidation. Rather, this acetyl coenzyme A will form ketone body in liver. So it will form ketone bodies. Ketone body. It will not go into Krebs cycle. So it will not be oxidized. It will rather produce ketone body. Ketone body for brain. It will produce ketone body for brain. This is very important fact. Just remember, we will require during ketone body synthesis. Now, so I have told you this. You can see this slide, pause this slide, and see how 1/6 I have